Good morning, everybody. Oh, forgot to put the lights on. Give me a second. Go. All right, a little bit more visible, which helps. Not that you really need to look at me anyway, but so today we are going to get onto control of gene expression in prokaryotes. Right, so first up, we need to talk a little bit about how prokaryotes do things, right? And so as we saw back, where be that? Here, back in translation, prokaryotes organize their genes into what are called operons. Let me get my chat box up. And really, genes that are involved in the same process as well. So like a metabolic pathway, for example, there's going to be a lot of different enzymes and proteins involved in metabolizing a particular substrate or producing a particular product. And so all of the genes involved in that process will be grouped together into what's called an operon. So all of the genes are going to be under the control of the same promoter. Right, so this essentially means that they coordinate their gene regulation for things that have to be produced together, right? This is called coordinated gene regulation. And eukaryotes do kind of this, get the same end, but they do it in a very different way, which we'll see later. And if you think about it, you want to make something. It takes multiple steps to do that, which making most things does, right? There's no point producing 3 out of 4 of those proteins required for that process because you're still not going to get anything, right? So you have to produce all of those proteins together, usually in similar amounts as well, depending on the situation, for that whole process to work properly, right? So it makes sense to kind of lump them all together. And so that leads on to the next point. So those genes are all transcribed into one long polycistronic mRNA. And what we mean by polycistronic is that it has multiple cistrons, and cistron is like a kind of fancy, not commonly used word to say open reading frame. So these are multi-open reading frame mRNAs, and each one has its own start and stop codon, and each one will be translated into its own distinct polypeptide and protein. Actually, a lot of viruses like COVID do the same thing. They have all of their genes kind of grouped together, and that generates one large mRNA, which is then translated into lots of different proteins. So in prokaryotes, the main focus of gene regulation is at the level of transcription, right? Because if you need something in terms of a metabolite, you have to produce or express the genes required for making that. If you don't need it, then you don't, so you turn it off, right? But it's all done all together, and it's mostly done at, I'm not an expert on bacterial gene regulation, to be honest. And so there may be some examples where it's not carried out at the level of transcription, but I'm not familiar with any. Let's go back here. And also, the region which is really kind of important in controlling gene expression is called the operator. And so keep in mind as well, right? You've got to keep in mind the whole way through the bacteria that if no proteins are bound, other than RNA polymerase and sigma factors and stuff, you'll get transcription. Right, so just bare DNA is wide open and ready for transcription. You have to turn it off by the addition of something else, another thing, in particular proteins. And those proteins will bind the operator. Okay, so I got kind of two sort of big concept type things to talk about before we get into the lac operon. One is how these operons are structured, and the other is the different levels or kind of coarseness, I guess, by which bacteria regulate their transcription. So let's look at the kind of structure of one of these operons, or like the, you know, the whole caboodle, right? All of the things together. And so essentially you have two parts, right? You have the operon itself, right? And this contains the structural genes, and structural genes doesn't mean these things are used to make structures. You know, it's that they're used to do things, like to make other things. Right, and it can vary what those are, right? So in the lac operon, you'll see that there are enzymes and there are channels and there's all kinds of different things, right? But these are the genes that encode the products that do stuff in terms of the purpose of that operon, right? Now, upstream of those structural genes, we have our regulatory region. We have a promoter. And so the promoter is where the RNA pol binds. And then we have the operator. And that's where regulator proteins bind. Now look to see where the operator is relative to the promoter. Let's kind of blow this up a bit. Right, so that'll do. Where the operator is relative to the promoter and to the structural genes. Right, it's in between. So what do you think would happen if a protein was bound to the operator? What would happen with regards to transcription? Or even RNA polymerase being able to start transcription of the structural genes? What do you think would happen? Right, exactly. It wouldn't be able to make its way through. And so this is in large part, not entirely, but in large part, how bacteria shut down gene expression. And this is at the very kind of on, off kind of deal, right? It's a very large scale, either works or it doesn't deal in terms of transcription. Okay, so that's one part, right? And these get transcribed into our polycistronic mRNA, each gets translated, and those proteins do whatever they're supposed to be doing, right? Now, the second part is a regulatory part, right? So the operon is transcribed, when it's allowed to be transcribed, right? It's not necessarily transcribed all the time. It very much depends on what kind of operon it is, right? What process those proteins are involved in. The second part is the regulatory part, right? And so this consists of another gene. It might be right next to the operon, might be way elsewhere in the bacterial genome. Doesn't really matter. And this gene is kind of always on, but at a low level, right? So it's always being transcribed, always being translated, and this regulator protein is always present, right? At low levels, right? It's something that's always needed, but you don't need a huge ton of it. This is the protein that, depending on context, right? And if we go back to here, come on, there we go. This is the context, right? So if that regulator is part of an inducible system, then it's going to bind to that operator in the absence of an inducer. If it's part of a repressible system, then it will only bind to that operator in the presence of the co-repressor, right? So the context by which this works or binds DNA very much depends on the context of how that operon is going to be regulated. All right, so you can't really assume one way or another without really knowing more about what's going on, the actual mechanism. But this regulated protein, depending on context and conditions, is what binds to the operator, right? So you can see that this gene doesn't have to be anywhere near the operator that or the operon, sorry, it's regulating, because this protein is diffusible, right? It'll be able to get there even if it's produced somewhere else in the cell. Okay, anybody got questions? Questions so far? This is a little bit of a hairy diagram to look at, but it'll make a lot more sense as we go through this. Okay. So the other main point that I wanted to make, so this is about like the structure, mechanics, I guess. The other point is really important to understanding how carefully, exquisitely almost, right, bacteria are able to control transcription. And so they use two kind of, I wouldn't say methods, but levels of control, right? So you've got what I call the Frankenstein's monster switch. That's like this big on, off, no Frankenstein's monster, right? So it's a major transcription is on, transcription is off, right? Big fold changes, right? You can go from very next to no transcription, and some of these, promoters are a little bit leaky, right? So even if a regulator is able to bind, sometimes it's not always bound, just stochastically. But essentially you go from next to zero to full chat, maybe 1,000-fold increase in transcription, right? So big difference. And then you've got like the dial, kind of rheostat almost. Actually, most of you probably wouldn't know what one of those is. You use like a Nest. There you go, modern equivalent, right? Kind of able to fine tune. I was like, yeah, no one's going to know what a rheostat is. Everybody knows what a Nest is, though, right? It's like this fine tuning, right? So in between the completely off, almost completely off, and gangbusters level, right? Often you need to kind of go somewhere in between there. And that's what that fine tuning is all about. And so both the examples that we're going to spend time going through have those two different levels of control, right? And that's really key to understanding how they work. And also, don't forget, right, we always talk about trying to get you to think about the big picture. You know, it's almost like, I'm trying to think of a good analogy. I don't know, it's like eating a meal when you're really, really hungry. Maybe that's not the best one, but it'll do, right? I've already started. So, you know, if you're absolutely famished, you kind of just scarf it down because you're super hungry, kind of barely even tasting the food. And then you kind of slow down a bit and just take it a little easier and just enjoy it until you're full. Right, that's the theory anyway. And so it's same for bacteria and control of an operon. You know, many times you will want to go flat out, right, until you have sufficient levels of whatever is down here, this product Y. And then once you get to the correct level, now you just want to maintain it, right? So rather than being like a life or death, we got to get this stuff situation, now it's like, okay, we got what we need, but we need to maintain that level. And that's where that fine tuning comes in. Oh, I wonder if we have time. Am I going to be able to find it? There we go. Let's see if we can. This is something from my youth. Let's see if this will actually. Ah, no, it's not. Crap. Okay, couldn't find it. Oh, there we go. Nick Park Creature Comforts. Let's see if we can find it there. No. Can't do a good job. Oh, I didn't even know they had an amoeba. All right, one day I'll have to dig it out and see if we can find that. It's basically a couple of turtles talking about how they like having just the right level of temperature, which I always think is funny. Anyway, that's a bit of a distraction. So it just occurred to me, random thought process. And so we're going to start with the lac operon because it's a relatively straightforward look into this. The trip operon is much more complex, right? So we want to kind of start you off on something a little bit more accessible. Okay, and so basically the lac operon consists of everything that's needed to metabolize lactose. And so you've got to bear in mind here for bacteria, glucose is the preferred food source. Basically, it's easy to digest. However, the glucose isn't always present, right? And so when it's not present, you have to use something, right? Because you don't want to starve as a bacterium. And so there are lots of other sugars, fructose, slightly more complex like dipeptides that can be broken down into glucose and something else, right? And so there are other sugars that can be metabolized if glucose isn't present. So if we're thinking about types of kind of control of gene expression, where would lactose fit, right? So we've got inducible and we've got repressible. Inducible is much more about things that you don't need very often, but when you do, you have to have lots of them. Repressible is more about things you need all the time, but you don't want to have more than what you need. So where do you think the lac operon would fit? Yeah, it's going to be inducible, right? Because you want it to be off when you don't need it. And when you do, then you want it to be turned on. Exactly, Isaiah, good job. So you want lac operon is an example of an inducible operon, inducible regulation. And so the lac operon consists of 3 genes. I don't know why we only have 2 there. There's the other one. Yeah, lac A, there we go. Right, which encode proteins that are responsible for the import of lactose, right? So lactose is a fairly large molecule, it's a disaccharide. So e.g. lactose is a disaccharide made up of glucose, where'd that go? And galactosides. Galactose pair. There you go. And proteins which split this apart, split lactose into glucose and galactose, and then glucose is metabolized. Not entirely sure what happens to galactose. Presumably it gets used somehow, but I'm not sure how. And we also have our regulatory elements, right? So if you remember back here, we have a regulator and we have a promoter and operator, right? In the lac operon, the repressor gene or regulator gene is called LAC-I. And then we have LAC-P for the promoter and LAC-O for the operator, right? So LAC-I is going to bind to LAC-O, essentially. Now, also, these 3 structural genes actually need to... let's just change this, because that'll bug me. There you go. One of the open reading frames in the operon encodes a lactose permease, right? So this is something that lets lactose into the cell, right? Because obviously you can't metabolize it if it's outside the cell, you have to have it inside. You also have the enzyme called beta galactosidase, right? And that's what cuts up galactose, well, lactose into galactose and glucose. And then you have LAC-A, which is not fully understood how it works, but it does something to the galactose. I'm not entirely sure. It's actually one of the least studied parts of this process. Okay, so this is kind of like the overview, essentially, right, pictorially. We have LAC-Y. Here's our permease, right? That's what allows lactose into the cell. I'm not sure entirely how it works. My guess would be it's some kind of active transporter, because this is bloody huge. Relatively speaking. And then that lactose is converted via one of 2 pathways using the same enzyme into galactose and glucose. Glucose then essentially heads into cellular respiration, gets broken down, turned into ATP. So this is the overview of what happens. Now we need to figure out, okay, well, how does this work in terms of regulation? Because we've got 2 things going on here. And this kind of comes back into that big kind of on-off switch and our on and off again dial, right? So first question, and this is arguably the most important one, is lactose present? Because if it's not, there's no point expressing the lac operon because there's nothing for it to metabolize, right? So that's the big, on-off deal. Lactose present, on. No lactose, off. And then the fine tuning deal comes from the amount of, come on, get back there. This present. So the amount of glucose that is present. And so if you think about it, the level of glucose depends on a bunch of different things. Is there glucose present in the environment? Right? If there is, you don't want to go through all the effort of producing beta-galactosidase and stuff because you just use glucose instead. Also, how quickly is glucose being used up by the bacteria? Right? If you have a very metabolically active bacteria, glucose is constantly going to be turned into carbon dioxide and ATP, heat, right? And then if you're on lactose, how much glucose is being produced, right? And so you want to have some way of figuring out how much glucose do we have on hand, right? Do we have a lot of glucose, in which case we really don't need much lac operon expression? Do we have a very little amount of glucose? Then we need more lac operon expression if lactose is present, right? And this is really what these two questions are what underpin the whole of the lac operon control. So keep that in mind, right? Keep coming back to that because that's the key to understanding this. And so the lac operon, like that big on-off deal, is controlled by the LAC-I repressor. And so that, just going to go back here, we've got our layout of the, and actually the repressor gene is right next to the lac operon. Again, doesn't have to be, but it is in this case. But the LAC-I, this repressor protein, will bind to the operator, LAC-O, in the absence of lactose. So when lactose is not present, LAC-I shuts down the lac operon almost entirely. There's a little bit of leakage because we need a little bit of that lactose permease for lactose to even get in if it is present, right? So it's not 100% off. It's not zero transcription, but it's very, very, very low. So this is again, an example of negative regulation, as is most regulation bacteria, because in the absence of LAC-I, you will get transcription of the lac operon. And so lactose is our inducer, right? And essentially what happens is when lactose is present, now we have a reason for expressing the lac operon. And that lactose binds to our repressor protein and prevents it from binding to the operator. Now the DNA is open, kind of open season, essentially. RNA polymerase will be able to bind to the promoter and then start transcription of the lac operon. That will give us our polycistronic mRNA and the polypeptides that will essentially get folded into proteins that will do the process that we looked at up here of importing lactose and then turning lactose into glucose and galactose. All right, so that's the main on-off switch. No lactose, lac-I binds, no transcription. Lactose present, lactose binds to lac-I, no lac-I binding, transcription can carry on or go ahead. All right, just give you a few seconds to absorb that. Anybody have any questions? Okay, well, go through this again, a little later. See if it kind of makes sense when I'm not in front of you explaining it. But again, keep in mind, really at the end of the day, what outcome does the bacteria want? If it was able to choose, or what does it need in terms of, survival and success? It needs to be able to use the nutrients it has available to it, right? And so it has preference for glucose because it's the simplest form of sugar. But if it can't find that, it's going to have to metabolize some other source of energy, right? And so lactose is just one of those. It's not the only one. There are lots of other different ones. So when lactose, essentially, it's almost like you have a series of kind of cookbooks or like manuals. It's like, well, normally I want to eat curry because I love curry, right? Any Indian food really, or Italian, whatever, right? But if I can't get curry, then I'll go, okay, what do I have in the fridge? I've got ingredients for lasagna. Okay. Pull off the recipe book. Obviously, don't need a recipe for lasagna. It's all up here. But then I'll use those ingredients and make lasagna. If I don't have things for lasagna, I go, okay, well, crap. I've got some eggs and I've got some bread and I've got some baked beans. Okay, baked beans on toast it is then. Pull off that particular instruction set. Right, so the bacteria has lots of these different kind of alternative energy source operons. But they don't need them when glucose is present, and they don't want to express them when the particular nutrient that they're able to metabolize is also not present. Right, so you've got to, it's like they're making a choice, right? Bacteria have a preference. If they can't get what they want, they'll see what's available, and then they'll kind of select whatever is the appropriate operon to be able to metabolize what's available. So until that happens, all of those other alternative energy source operons are off. And the thing that turns them on is the metabolite or nutrient they can use, right? Their substrate, essentially. And so those are all inducible, and they're always going to be induced by the thing that they're able to utilize. And so in the case of the lac operon, it's lactose. Yeah, so kind of keep that kind of bigger picture in mind, and then these things will slot in quite nicely into that picture. Just in and of itself, it's a lot of just stuff, right? It's hard to remember. Some people are able to do really well with that kind of information, others less so. Always like kind of, how does this fit into this particular picture? And obviously, terrible and not very accurate analogies as well is kind of another one of my skills. Okay, but remember we're talking about, okay, this is like the big on-off switch. Now, at some point after, bacteria finds that, You know, glucose isn't around, but lactose is, and it needs glucose because its glucose stores are very low, then it will go full on out to make these various proteins that turn lactose into glucose, right? So it kind of replenishes its stores essentially. And then once the level of glucose starts rising, right? And again, you know, bacteria can't store huge amounts of glucose, right? It's not like, you know, eukaryotes like us, where if we have excess glucose, we just turn it into fat and store it around our guts or our butts, right? Bacteria can't do that. And so they have to kind of very carefully modify how much of something they're producing, because, you know, they can't have too much of it essentially. And so when glucose levels start rising, now we need to dial back expression from the lac operon. So we just don't need as much lactose being turned into galactose and glucose. Even if there's still an absolute ton of lactose present, right? Essentially, you know, the bacteria is getting full. And so this is where we come to the fine control. And this is all about sensing glucose levels. And so glucose cannot be directly detected, so, or glucose levels. And so it has to use kind of like an intermediate sensor, I guess. And this is also something that we do, you know, so when your glucose levels drop, essentially there's another metabolite called cyclic adenosine monophosphate, or cyclic AMP, which rises. And so cyclic AMP is kind of like a starvation sensor, right? That's really let's just type that out before I forget. So when your cells, you know, say your muscle cells, if you've been exercising, start running low of glucose, cyclic AMP goes up. And that tells those cells to either increase import of glucose or to essentially turn any stored carbohydrate like glycogen or not so much fat, but other stuff into glucose so it can be used, right? So it's the same kind of deal. Essentially, when glucose is down, cyclic AMP is up. And when glucose levels increase, cyclic AMP levels drop. Right, so the thing that the bacteria is detecting is the level of cyclic AMP. Okay, so that's the thing it's detecting. The question is how? How do bacteria detect, in the case of the lac operon particularly, levels of glucose, right? The thing that's being produced, right? So again, this is kind of comes back to that whole like, I don't know, auto-regulation kind of deal, essentially. And so that protein is called the catabolite activator protein. And this is where it gets a little bit loony. So we can kind of refresh ourselves on this next class just to make sure it sinks in, because this is not kind of a trivial thing to get. Okay, so we want to know how much glucose is present, right? We detect that by sensing levels of cyclic AMP, right? So if glucose is up, cyclic AMP is down. We detect cyclic AMP by the catabolite activator protein. And so how do we do that? Maybe the next question on your list. All right, so basically CAP plus cyclic AMP equals more efficient transcription. So when glucose levels are low, and lactose is present, remember that's the kind of the main on-off deal, right? If lactose is not present, this doesn't matter. You're not going to get transcription of the lac operon, right? But if lactose is present and glucose levels are low, CAMP binds to CAP plus cyclic AMP helps RNA pol to bind to LACP promoter equals higher levels of transcription. Oh, that little arrow's there for. Now you're going to produce a lot of the products of the lac operon. You're going to be turning lactose into glucose and galactose. And now glucose levels are going to start increasing, assuming the same level of glucose consumption, right? And so now you have a lot of lac operon expression equals lots of lactose being turned into glucose. So glucose levels increase. So if you kept going at the same rate in terms of lac operon expression, you'd still get ever more levels of higher, more and more glucose, right? And you don't want that, because transcription and translation is expensive and you don't need more glucose. You have enough. You just need to maintain that level of glucose, right? So you can do whatever one needs to do as a bacteria. So now you need to start dialing back transcription from the lac operon. You don't want to turn it off because you still have lactose present. You just don't need as much of it. So as glucose levels increase, cyclic AMP levels decrease. Remember, as one goes up, the other one goes down, and vice versa, right? Glucose is down, cyclic AMP up. As glucose increases, cyclic AMP levels drop, right? So there's less CAP plus cyclic AMP. So RNA pol binds less efficiently, equals lower but not none levels of lac operon transcription. So it does happen. It just doesn't happen at the full-on levels that we had earlier on, where we have lactose but no glucose. Right, so essentially you've got kind of two extremes here, right? So one, glucose is present and lactose is absent. And that equals no lac operon expression. Because you don't need it, right? Glucose is there and there's no lactose. Or even if glucose isn't there, if there's no lactose, it doesn't matter because you're not going to have any lactose to turn into glucose, right? Some other kind of alternative nutrient source operon would be turned on. And at the other extreme, glucose is present and lactose is present. Or actually, glucose is absent, sorry, and lactose is present, equals very high levels of lac operon expression. Right, so you don't have your preferred source, you do have an alternative source. We got to make the use of that and make lots of glucose. Now, in between, you have an intermediate level of expression, right? Not the zero if lactose is absent, not the like full on all hands on deck level if glucose is absent but lactose is present. Now you have something somewhere in between, right? And that somewhere in between depends on the levels of glucose. Right, so lactose is present so the lac operon can function, but glucose is also present so you don't want it to function very well, right? Right, and so depending on the level of glucose, right, which determines the level of cyclic AMP, will determine where between those two extremes you have expression for the lac operon. Right, obviously some glucose, but not a lot, you'll still get quite a lot of lac operon expression. Lots of glucose, you'll get very little lac operon expression. So that's the dial, right? Where you figure out where in between you want that level of expression to be. As I said earlier, it's a very elegant way of doing things. All right, that's enough for now. We'll go through this, see if it makes sense. Bring any questions to the start of the lecture on Friday, and we'll have a bit of time to go through this before hitting the trip operon. And the trip operon is pretty nuts, right? So it's more complex than this. So you want to be kind of loins girded and fully prepared, essentially, before we hit that. All right, thanks very much, everybody. I shall see some of you in lab in a little bit, and the rest of you, have a lovely day, and I'll see you on Friday.